Hello, my name is Alexandra Eakes. I'm a Princeton University student, class of 2017. I'm part of the Ecology and Evolutionary Biology department, as well as pursuing a certificate in global health and health policy. So I decided to do my independent research project on the risk of Chagas disease in the Caribbean. My project is in collaboration with the University of the West Indies in Trinidad, as well as St. George's University in Grenada. I chose to focus on the Caribbean because though Chagas disease is largely endemic in much of South America, there isn't much research in the Caribbean. So I wanted to know, is there a risk for Chagas disease in Trinidad?
Chagas disease is the infection of the parasite called Trypanosoma cruzi in a human. So what happens is when you're first infected with Trypanosoma cruzi, you might get indescript symptoms that seem like the flu. But then you never essentially get rid of the parasite. And so that over 10 to 30 years, the parasite is slowly multiplying inside your body. And the tissues that the parasite prefers are cardiac tissues and gastrointestinal tissues. And so after 10 to 30 years of the parasite repeatedly multiplying and repeatedly triggering inflammation in those tissues, this can result in severe cardiac or gastrointestinal problems, including death.
Trypanosoma cruzi, the causative agent of Chagas disease, is transmitted to its mammal host through the feces of its vector, the triatomine bug. Currently, there are 6 known species of triatomine bugs on the island of Trinidad. The aim of my project was to collect these triatomine bugs and determine what is the prevalence of Trypanosoma cruzi in these bugs, as well as are they making contact with humans. Noiro traps hold a mouse inside and are wrapped in super sticky tape. The bug is attracted to the scent of the mouse, thinking it will get a blood meal, and gets stuck to the tape. One of the places traps were placed was in the nesting sites of potential mammal hosts. One of those locations is in cocoa-reed trees as shown here. Others were placed in holes in the ground where animals might burrow at night. Other traps were placed near old abandoned homes or near the porch lights of current homes, as the bugs are attracted to these lights at nighttime. When a bug was caught on the trap, the next day we would remove the bug from the tape and put it in a collection cup before taking down the trap for the day. Collection cups were also given to locals in case they found any in their homes. After the feces were examined under the microscope, looking for the parasite, the DNA was extracted. This DNA was then used to perform Trypanosoma cruzi PCR. This was used to determine the prevalence of the parasite among the bugs collected. Next, a blood meal analysis was performed to determine the last blood meal on which the bug had fed.
So blood meal analysis. So the neat thing about PCR is that you can pick up DNA targets of a variety of different types, even within a single sample. And so what we were doing here with our time is we were doing some analysis of Trypanosoma cruzi in the samples. So you crush up the bug, and then all of the DNA will be, all of the DNA that's inside of that, everything from the bug itself, whatever host it was feeding on, or any pathogens that are inside of it will all be inside of that tube of DNA. So what we did was we used Trypanosoma cruzi specific primers, which Allie did prior to me coming here, or to her coming to me rather. We did PCR using vertebrate specific primers, which would pick up neither the pathogens nor the insect host. It would only pick up DNA that was still in the bug from the meal that they had fed on. And so that helped us figure out what they were feeding on. This gives us an idea on an epidemiological level of what the bugs are feeding on and what they may be transmitting the disease Trypanosoma, or the pathogen Trypanosoma cruzi to.
All of the bugs collected were found in and around human homes. Of the bugs collected, 84% tested positive for Trypanosoma cruzi. The blood meal analysis work is currently ongoing. However, a few of the sequencing results have come back, and 3 of them have been positive for human DNA. So one of the bugs actually that was positive for human DNA was also positive for Trypanosoma cruzi. And so this is important because it indicates that the vector infected with the parasite is coming into contact with humans. This poses a public health concern because it indicates that Chagas disease could be transmitted in the Caribbean and is possibly currently underdiagnosed. It is something that health officials should be made aware of and is something that should be further studied.